Hi, I am Mrs. Sloan, and this is Cell Structures and Functions from my Honors Biology class. This is part 2. We have already reviewed, for instance, the cell theory, so you might want to go back and look at that if you haven't looked at that already. We've talked about why cells are small. We have also discussed the structure of prokaryotic cells and the components and what each does. Remember, prokaryotic cells contain no membrane enclosed organelles. And we also did a little overview of the 3 domains and the 4 kingdoms we discuss in the eukaryotic domain, all right? So we're all about part 2 now. And let me get in presentation mode. Oh, there I am on top of myself. Okay, here we go. So we are in the notes, which are down in the descriptor of this video. My students do 2-column notes, and the first column is scaffolding of what I'm talking about, and then there's little blanks that are yellow highlights. You want to type those in. And the second column is add in pictures and things that are helpful for this presentation. All right. So I am on Introduction to Eukaryotic Cells. And it says in general. So the big thing you need to know about eukaryotic cells is they contain membrane enclosed organelles, specifically a nucleus. I see if you can kind of see it there in the middle a little bit. Let me get myself a pointer. All right. So you can probably Google a great picture to put into your notes with structures and functions. This is a typical animal cell. They're all different depending on what their function is, where they are located in your body. But you can see this one has all the different parts identified and what each does. So let's just get our bearings, like an overview of this real quick. So we can see the nucleus here in the center. This dark part inside the nucleus is called the nucleolus, and I'll address that in just a minute. Going immediately around the nucleus and continuous with the nucleus is called the Endoplasmic Reticulum. There's rough, it has little bumps on it. Those are ribosomes, and then they're smooth. And then the nucleus and the ER are very well connected. And then next would be the Golgi apparatus. These 3 are all connected with each other, and we'll talk about that. And then you have the powerhouse of the cell, mitochondria over here. Notice there's no chloroplast because it's not a plant cell, and there's no cell wall and no large central vacuoles, but you'll have smaller vacuoles. Little things over here that look like churros, and lysosomes are like the stomach, and of course the phospholipid bilayer. So we're going to learn these different parts and also what you would see in a plant cell as well. So in general, that has, number 1, membrane-bound organelles, like a nucleus or mitochondria. That's in your notes, right? Number 2, it's larger. So because it's larger, right, it has less surface area to volume. Remember we said the smaller the cell, the larger surface area to the volume. Since this is larger, it has less surface area to volume. You know how it makes up for that though? All the compartmentalization within the cell, all these membrane enclosed organelles. So number 3, it's compartmentalized. It has organelles that perform certain functions. Remember, prokaryotic cells, one cell needs to take care of everything. Here they have subdivided the responsibilities into different organelles. All right, okay, here we go. So we're going to start with the brain of the cell. Now here is an electron microscope. You've got a micrograph. You've got you can see the nucleus here is dark purple, then the nucleolus, and then surrounding it is the endoplasmic reticulum. When you look inside the nucleus, you see how there's different stains inside that. Inside your nucleus, you have mixtures of DNA, there's some RNA in there, there's some proteins in there, and heterochromatin, dark staining DNA is like tightly coiled and put away, and euchromatin is the active one that's lighter whenever you look, okay? So it's a mixture when you look inside. Let's zoom in a little bit closer, okay? So here, you can see the nucleus, you can see the nuclear envelope. Think about this nuclear envelope right here. The passageways, these nuclear pores, are going to be super important, right? Because inside you have your genetic code book. And think about your phone, right? Your phones probably have a passcode or something or facial recognition to let them in. These nuclear pores are very protective of the genetic code that's inside of it. Now, one of the items that is made, when the DNA is the information, how you display that information is via proteins, right? And to make proteins, you need ribosomes. And ribosomes are built out of our RNA. Remember DNA, RNA, ATP, the 3 types of nucleic acids. And this is where that's getting done right here. Because you need a lot of ribosomes, that's occurring right there in the nucleolus. Now, if you've ever already had an introduction to mitosis or meiosis, you know that DNA can coil itself onto rod-shaped chromosomes that look like this. Let me show you a couple pictures and then I'm going to come back there. Okay, here's your DNA double helix. It twists up on these histone proteins. These right here are called nucleosomes. It's labeled right here, nucleosomes. Okay, and it winds up, and organizes itself into a chromosome inside the nucleus when it's getting ready for cell division. So if you've ever seen pictures like this, this means it's in the middle of mitosis or in the middle of meiosis, all right? So let's go back here and look at our nucleus and go on to our notes. So nucleus, number 1, chromatin or chromatin is the thread-like material that coils into chromosomes. The thread-like material that coils into chromosomes. Chromosomes are rod-like structures formed during cell division. This red item right here, this is a chromosome. Rod-like structures that form during cell division. The nucleolus right here, the dark part in the center, nucleolus, is where you have ribosome formation. Ribosome formation. And then the nuclear envelope going around the outside has nuclear pores to permit passage of substances in and out of the nucleus, in and out of the nucleus. And if I was going to give a human body part to the nucleus, you know what I'd call it? I'd call it the brain. It is the brain of the cell, okay? It is the CEO of the cell. So let's talk about ribosomes. Since ribosomes are made right here, let's look to see the significance of a ribosome. Now, we're going to have a whole chapter on this, so don't stress out. Okay, so ribosomes have a small and a large ribosomal subunit, and proteins are built on ribosomes. They are the workbench for protein synthesis. Long story short, DNA is your code book. You can make a copy of the leading strand of DNA, and you can make an mRNA copy. That mRNA will go out of the nuclear pores, and once it goes out of the nuclear pores, it'll get on a workbench, a ribosome. And then you have things like tRNA that are transferring amino acids because, amino acids hooked together will build a protein, right? Do you remember what kind of bonds are between those amino acids? Did you say peptide bonds? Yay, peptide bonds formed through dehydration synthesis. So the building of the primary structure of this protein actually occurs on a ribosome. And ribosomes are really small. And what can happen is the mRNA can be there and a ribosome can move right along it and you can make a protein and another one. You could have a whole line of ribosomes, a polyribosome, just working that mRNA, building whatever that protein is. So on your notes, ribosomes, workbench for protein synthesis, composed of 2 subunits, small and a large subunit. Perfect. Okay. Check. Let's move out. Now, the next structure we're going to look at is the endoplasmic reticulum. And I want you to know the endoplasmic reticulum, the membranes of it are connected with the nuclear envelope. So you can't really see, it just is continuous, right? Where a nuclear envelope is, then all of a sudden you can see the endoplasmic reticulum is starting. And what I want you to think of this is think of it as a twisted road through the cell. Okay, a twisted road through the cell, and it's a membrane sac. And some of this twisted road has ribosomes on the outside, and that's why it's referred to as rough ER, because it looks bumpy because of the ribosomes. So you know what's happening on those ribosomes, right? Proteins are getting made. Proteins are getting made. And then there's smooth ER. Smooth ER are not about building proteins. They're about building lipids. Okay, now what I want you to see, if you can see, right here, nuclear envelope, look right here. Here's a nuclear pore. So this literally, that mRNA that you make or ribosomes that you make come through the nuclear pore, this mRNA, and it can get right here on one of those ribosomes so you can start building proteins. And then what will happen is when the protein is built, it'll move right inside of the endoplasmic reticulum. So you can build a protein here. It'll move right inside of this endoplasmic reticulum. And what that endoplasmic reticulum will do is, remember how we learned about secondary, tertiary, quaternary structure? It starts to fold it. It might manipulate it. It might snip it and trim it around a little bit. And then what will happen is, because this is just membrane material, it will just bud right off of the rough ER. So now your protein is packaged in a membrane material, and it's called a vesicle. Okay, so let me hit that one more time because I hit it kind of hard, all right? So mRNA could come out of the nuclear pore, get right here, the messenger RNA, a copy of the DNA, get on this ribosome, a protein is built, the protein will move right into the interior of the ER, it'll get modified, folded, and then it'll get released out the end of the ER. So that's the rough ER. So let's go to our notes. First of all, this whole endomembrane system that we're going to be talking about, the rough and smooth ER, the Golgi apparatus, and the lysosomes, that whole endomembrane system in general, it's a series of intracellular, within, right, intracellular membranes that compartmentalizes the cell. The endoplasmic reticulum is connected to the nuclear envelope. The rough ER has ribosomes for protein synthesis. You already have that. The proteins enter the ER for processing and modification. So the smooth ER is connected. You can see right here, here's rough ER, and then all of a sudden it becomes smooth ER. The smooth ER is about lipid formation, manufacture, and release, and also it is a detoxifier. So for instance, if you made the poor choice of getting too much alcohol in your body, either in one setting or in multiple settings over time due to alcoholism, your smooth ER are going to be working to take care of that alcohol, to detoxify it. And what happens with alcoholics is that their smooth ER gets destroyed in their liver cells, right? And then their liver cells die and it gets replaced with connective tissue. And then that's not good because then your liver can't do the job of your body that your body needs. So on smooth ER, it's connected to the rough ER. It doesn't have any ribosomes on it. It forms lipids, which you know are fats, right? And it's in charge of detoxification, storage, and forms vesicles. All right, let's keep going. Here's a really good picture. It looks like all-seeing eye. You can see the nucleus. You can see how that rough ER just envelops it because it's continuous. And then you have your smooth ER. All right, here's another picture, rough, and then you're into smooth. All right, now, continuing on our endomembrane system, whatever is made in the smooth ER or the rough ER, right, it then gets shipped to the Golgi apparatus. And the Golgi apparatus, I like to think of as the UPS man of the cell. Like if I brought you that, I could take this to the UPS store. I can't just mail it just like this. They've got to put it in a box, make sure it's all protected, put an address label on it, and ship it. Maybe it's going to go somewhere here in town. Maybe it's going to get shipped to Europe, right? The Golgi apparatus takes care of that. He is your UPS man. So what happens is if it's coming from the smooth, look up here at the smooth ER, transport vesicle, it'll go into the Golgi apparatus. By the way, the cis side faces the nucleus. The trans side faces away from the nucleus. And if it's coming from the rough ER, okay, it will still next leave in a vesicle and then dock with the Golgi apparatus as well, and then be transported. It could be transported completely out of the cell. When the Golgi apparatus is done, maybe it's holding insulin and you need to secrete that into your bloodstream. So it could go all the way to the edge of the cell and release it just like that. Okay, and that would be a form of what? Do you remember? Not endocytosis, but exocytosis. Good. So on your Golgi apparatus, the UPS man, it receives vesicles that bud from the ER, either rough or smooth. Lipids and proteins are sorted and packaged. Some will exit the cell, discharging secretions through exocytosis. All right, let's talk about one that's not going to leave the cell. Oh, yeah, the lysosome. Lysosome is like the stomach of your cell, okay? It has in it strong hydrolytic enzymes. Ooh, hydrolytic, hydrolysis, breaking, right? It has enzymes in it. And what happens is it will fuse with a vesicle that has nutrients in it and then digest those nutrients up. So let's look at an example of a lysosome, okay, and how that works. Let me put myself up over here. Okay, so start down at the bottom, this summary, okay? Here's your nucleus. You can see the rough ER. You can see some proteins in here, and it leaves the rough ER in a vesicle. It goes to the cis side of the Golgi apparatus. He's further modifying it, packaging it, and in this case, this is a secretory vesicle. It's getting completely released. Let's look at another option. Another vesicle right here, this lysosome, has hydrolytic enzymes in it. Remember, enzymes are usually proteins, so this probably came from the rough ER, right? And this lysosome is going to merge with this vesicle. Look what's happening here. This is cell eating. What do we call that form of endocytosis? Do you remember? Did you say phagocytosis? Yay. Okay, so it's engulfed this food and it's going to merge with the lysosome, the stomach of the cell, and it's going to digest the nutrients inside of it and absorb whatever it wants to absorb, and then it'll move to the edge and release any waste, okay? Just like you do egestion, right? Going poo. All right, now, the lysosome does other things besides just digest food. Sometimes some of the organelles aren't working, aren't functioning anymore. So the lysosome will fuse with a bad organelle and destroy it. So you can repurpose its parts, right? You can repurpose the monomers. Also, sometimes you just need to have cells die. Do you remember what that's called? That cellular suicide is apoptosis. So what a lysosome will do is just release its hydrolytic enzymes to the entirety of the cell, and it just kills the cell off, all right? So lysosomes contain powerful digestive enzymes that are highly acidic, and they fuse with vesicles digesting their contents. Examples, white blood cells and bacteria. Like if a white blood cell eats a bacterial cell, it'll destroy it. Autodigestion of parts of the cell, like if you're going to destroy a part like a different organelle, and apoptosis. And what you want to put in the parentheses after apoptosis is programmed cell death. Programmed cell death. So that's what you refer to as the endomembrane system. Okay, you've got your nucleus, then your endomembrane system would be like the rough and smooth ER, the Golgi apparatus, and any lysosome that you would form. Now, there's some other vesicles too we want to learn about, okay? One is a proxisome. A proxisome, they are made not on ribosomes associated with the ER, but on ribosomes that are just in the cytosol, the fluid portion. And proxisomes are, like, what do you hear in that? Proxisome, peroxide, right? They are a detoxifier as well. And they break down some of the products that we generate through our metabolism. So on proxisomes, specifically, they break down fatty acids. Breaks down fatty acids. Now, side note, which you're going to add to your notes, but side note, if you've ever gotten a cut, and what do you put on that cut? If you put on hydrogen peroxide, it'll bubble, right? And that's because inside your cells, in your proxisomes, you have something called catalase. And catalase will break down the hydrogen peroxide you put on your cut into its two components, water and oxygen. And it's the oxygen that bubbles that you see. Now, hydrogen peroxide will do that on your tissues, but if you chopped up some pieces of raw potato and put hydrogen peroxide on that, it would bubble as well, because eukaryotic cells have proxisomes with catalase in it. Why do they have it? To take care of things like a buildup of hydrogen peroxide, which would be a byproduct of metabolism. Now, why do we put it on our cuts then? Because the hydrogen peroxide is toxic to the bacteria that might be infecting you, but I'm just telling you how your body responds to that toxin by breaking it down with catalase. So you want to add on there to proxisome, contains catalase, C-A-T-A-L-A-S-E, contains catalase, to break down hydrogen peroxide. And if you need to, after that, put product of metabolism. Okay, good job. Next, let's look at vacuoles. Now, We have vacuoles in our animal cells, but they're super big in plant cells. And the reason why they are large central vacuole in plants is because they're like giant water balloons. They fill up with water, and when they expand, they push all the organelles and all the cytosol up against the cell wall, and it helps keep the plant cell rigid and upright so it can absorb the sunlight for photosynthesis. Other vacuoles could be digestive vacuoles or for storage as well. So on your notes, vacuoles, large membranous sacs. It is known as the multi-purpose organelle. If I was going to pick a building like on campus, this would be like, oh, I cannot remember the name of it. Like a multi-purpose room where you put on plays or you go to meetings. That's what the vacuole is, kind of a multi-purpose structure like G9 or something like that. Or I can't remember at Oak Park. Somebody's got to tell me. I'll think of it. It'll be in that puzzle. Anyway, so that's your multi-purpose room vacuoles, multi-purpose organelle. Central vacuoles in plants. So central vacuoles in plants maintains turgor pressure. So maintains turgor pressure in plants. Contractile vacuoles in protists. Protists is a type underneath the domain eukarya, usually unicellular, but eukaryotic cell. So contractile vacuoles in protists are for water regulation. Water regulation. They literally squirt out water because they live in a hypotonic environment. And you can have digestive vacuoles and storage vacuoles. Yay. Okay, so next, let's talk about our energy organelles. And we have a chapter for each one of these. They're so important, okay? So plants have chloroplasts, or any autotrophs, photoautotrophs, right, have chloroplasts. The product of that is sugar, right, and oxygen. And the powerhouse of our eukaryotic cells, mitochondria, we need sugar and we need oxygen, and we oxidize that sugar and oxygen into ATP. And ATP is the cheddar, the energy currency of the cell, and the mitochondria makes that for us. Now, mitochondria are found in all eukaryotic cells. So don't say plants have chloroplasts and animals have mitochondria. No, that's an ignorant statement, okay? Photoautotrophs are going to have chloroplasts, and all eukaryotic cells have mitochondria. It's a powerhouse of cell, and all those cells need ATP. So they will generate ATP. Now, the byproduct of cellular respiration, like think about it right now. I'm breathing in, breathing out. What gas am I giving off? CO2, right? CO2 and water, the products of cellular respiration, are the reactants for the chloroplast and vice versa. So we're going to talk more about those later, but I want to address, so you can skip down in the notes where it says 4.7, the energy organelles. Chloroplasts are the site of photosynthesis. Okay, and mitochondria, if you jump down to that, the function is cellular respiration. Now, I want to tell you something pretty cool about mitochondria and chloroplasts. First of all, it's the origin of eukaryotic cells. We studied last time about prokaryotic cells. They have no membrane enclosed organelles. How could eukaryotic cells have evolved? Long story short, two ways. One prokaryotic cell went and engulfed another prokaryotic cell. Okay, so that's part one. These mitochondria and chloroplasts could have been free-living prokaryotes at one time, and they just got engulfed but not digested by another prokaryotic cell to make it do its work for it, to make it make sugars or to make it make ATP for it. So that is called the endosymbiotic hypothesis, the endosymbiotic hypothesis. So this could be a prokaryote, and it would engulf some aerobic, other prokaryote to make it be its mitochondria, and then engulf a chloroplast, if you're a plant cell, to make its sugars for it. Okay, so that would explain mitochondria and chloroplast, and I have more information about that. Just put a pin in that. But then how would you get the nuclear envelope, right? How would you get the whole endomembrane system? And I want you to think about endocytosis, right? Endocytosis is that folding in. So what if a membrane folded in, but it folded in a lot? So it became like a little ribbon highway I was talking about earlier. And that ribbon highway made its way around the nucleoid region, right? The region where the DNA is. and became then a nucleus because it had a membrane barrier around it. How could that have happened? Fitness. The best cells survive and reproduce. And those that protected, so you had a infold of membrane around the nucleoid region to make a nucleus, those that protected it were probably the ones that survived and reproduced. And then that leftover ribbon would have been what's continuous now, the rough and smooth ER. Part of it could have broken off to make the Golgi apparatus. So take a look right here, okay? And, oh, I'm one of the cells. Let me go over here. On the endosymbiotic hypothesis, okay, right here. So how could you get this DNA covered? Well, you could have had infolding, okay? So see where it says infolding, invagination in? This could have folded in and worked its way around the DNA to form the nucleus, and then engulfing either aerobic bacteria to be an animal cell, or engulfing that aerobic bacteria and additionally some photosynthetic bacteria to be the chloroplast. What's the evidence for that? Really interesting. Both mitochondria and chloroplast have their own DNA. And guess what? It's single circular chromosome, just like in bacteria. They have their own ribosomes. They can make their own proteins. And they are self-replicating by binary fission, which is how prokaryotes replicate. Also, they're double membraned. So what I mean by that is there is, look right, let me get one. See how there's an outer and an inner membrane right here, and then some other inner membranes in here. The outer membrane is like eukaryotic type membrane from the engulfing, and the inner membrane is more structured like prokaryotic cells. So that's where that's all the evidence. So let's write that down. So in general, on the notes underneath 4.7, the endosymbiotic hypothesis states that these two organelles may have once been free living bacteria that went to live in other bacteria. This could point to the evolution of eukaryotic cells. They have a double membrane, possibly picked up the second membrane as it was engulfed. They contain their own DNA, ring-like structure as in prokaryotes, and their own ribosomes. They can make their own proteins. And number 3, they are self-replicating. All right? Now, chloroplasts, they are the site of photosynthesis. Now, I skipped over one slide. Don't stress about it. The bigger umbrella on this is plastids. So chloroplasts are a type of plastid, a colored one, leucoplastore. But we're not going to worry about that right now. Okay, but chloroplasts are the site of photosynthesis. We'll have a whole chapter to talk about that. Okay, here is a picture of a chloroplast. Look how large it is in relation to the central vacuole. This is your nucleus over here, and this part of the mitochondria. Okay, and then here is mitochondria. You can see the double membrane, right? Just like I talked to you about before. Mitochondria, the function is cellular respiration. And compared to chloroplasts, they are smaller and more varied in shape. And then, ironically, just a little bit of trivia for you, all the chloroplasts in your body right now all came from your mother. All came from your mother. And we'll talk about that part later. But diseases, there are over, ready for this, 40 different mitochondrial diseases. 40 different mitochondrial diseases. And remember how I told you your mitochondria has DNA in it, right? So if you analyze the DNA in your mitochondria, in your cells, that's the same DNA from your biological mother and from her biological mother and from her biological mother. You can see where I'm going on that, right? You can trace your mother's lineage DNA in her mitochondria back. All right? Last big topic, okay, we need to talk about is the cytoskeleton. Just like you have a skeleton in your body for structure, support of your body, for protection, cells have cytoskeletons as well. And you can kind of see it outlined there in the background. Okay, here's another one. So I don't want you to think that these organelles are just floating randomly around inside a cell. They are literally supported by different aspects of the cytoskeleton. So it is, on your notes, a network of connected filaments and tubules, a network of connected filaments and tubules that extends from the nucleus to the plasma membrane. So it extends from the nucleus all the way out to the plasma membrane. Now, just like you have big, middle, and large bones, like your femur is the largest bones, it's the same thing for cells as well. The tiniest ones are actin. It's like two chains of amino acids twisted together. Intermediate, several chains twisted together. And then microtubules, They are the biggest structure, but it's not so much because they're a solid wire that's twisted together like that, but because they make it like a hollow cylinder, and these are called microtubules. So on your notes, actin filaments are thin fibers, and they form a dense web right underneath the cell membrane, okay, right on the edge of it. So thin fibers that form a dense web of structural support under the cell membrane. Intermediate filaments, I gave you everything around about that. They support the nuclear and plasma membrane and part of cell junctions. And then microtubules are hollow cylinders that can act as a cellular railway, So here, I want to just point out to you muscles. When you contract your muscles, two of your cytoskeletons are sliding past each other in your muscle fibers, specifically in your sarcomeres. So you have thin actin and myosin, and it literally grabs the actin to contract your muscle, like if you're trying to make a muscle, okay? That's one place. And then here, you can see that the microtubule here, okay, this vesicle, like say it's an endocytotic vesicle, is literally getting walked along the cellular railway of these microtubules right here. All right, then this leads us to the churros, as I like to call them, but they're called centrioles. You only see them in animal cells, okay, you don't see them in plant cells, but can you see how there's, if you count around, there's 9 groups of 3. Those are microtubules that make up those centrioles. What are they for? Don't know. Okay, because plant cells do just fine without them, but I can tell you where they're located. There's always 2, and they're perpendicular to each other, but when you get ready to do cell division, they sit right in the center of where this either end of the cell when it's dividing. It's called a microtubule organizing center or a centrosome where they sit. But apparently you can do cell division without them because plants do, but animals do have them. And there are 9 groups of 9 groups of 3 microtubules. So on your notes, put centrioles, and next to centrioles, put 9 triplets. You're going to have to type that in, 9 triplets. So the structure is short cylinders found in pairs in animal cells and most protists. Remember, protists are one of the kingdoms underneath the domain Eukarya. Okay, now, interestingly enough, where you see that same structure is not at cilium flagella, but where you anchor the cilium flagella. And I'm going to show you another picture of that in just a minute. So skip over basal bodies for right now and go to cilium flagella. Cilium flagella are all about movement, okay? Cilia are a lot of little short hairs like what you're seeing right here. And those are either to move like a protist to swim around, but like you have cilia in your trachea that help bring up any pollutants that are in your trachea, and so you cough and spit those out so they can also move fluids. Flagella, like you see in the sperm in the top picture, that flagella helps with movement of a single cell, okay? Now, cilium flagella are all about movement, so put that next to it. And what I want you to write after movement is 9 doubles around 2 singles. Okay, so if you look right here, instead of triplets, it's 9 doubles of microtubules around 2 singles. And you can see that in this picture here. Now, okay, that's how cilium flagella are built. Now, what anchors cilium flagella is called a basal body, but basal bodies look just like little centrioles. Okay, so their structure, so here's centrioles, 9 groups of 3 right here, 9 groups of 3. Here are what cilium flagella are made out of, 9 groups of 2 and 2 singles. And here's a basal body. The basal body is what anchors the cilium flagella right down at the bottom, okay? And those have that same structure of 9 groups of 3. So where you have basal bodies under centrioles, same structure as centrioles, and may have originated from them. They anchor cilia and flagella. And then you have cilia is shorter, usually more, and can have substances across their membranes. And then you have flagella is longer. And flagella is longer. Okay, good. All right, my friends, listen, this is the end. This is the end of this, but I want to highly recommend that underneath at the bottom of your notes, okay, that you go find some great pictures of plant and animal cells, and if you haven't already, a prokaryotic cell that's labeled and identified, and put that at the bottom of your notes so you have that as a review. Okay, I'm proud of you, my resilient children, and that's all.